Hello, dear BSC students. We have already finished with Plasmodium vivax. We have seen the geographical distribution, habit and habitat of Plasmodium vivax, its vectors, its life cycle, its effects on the host, and then at the end we have seen the prevention measures of the Plasmodium vivax. So now we will continue the next part of this chapter, that is Entamoeba Hystolytica. This is another type of parasite that we are going to study. So we will start with the systematic position of Entamoeba Hystolytica. So as you can see, it belongs to the kingdom Protista, subkingdom is Protozoan, phylum Sarcomastigophora, subphylum Sarcodina, class Lobosi, order Amoebida, family Entamoebidae, and then you have a genus Entamoeba and species Hystolytica. There are three species in within this genus. So Entamoeba Hystolytica, Entamoeba coli, and Entamoeba gingivalis. But in this chapter we are going to study only the Entamoeba Hystolytica. Now if you look at this, the genus Entamoeba, it includes at least 20 species, and all in various invertebrates, vertebrate hosts. They live in vertebrate and invertebrate hosts. Remember, they don't live outside. Yes, some part of them they are present outside, but nourishment and development that doesn't happen outside. So basically they are adapted for the adaptation is related with the any host. So without host they cannot survive. Out of all these three species whatever we have talked about, out of that Entamoeba coli and Entamoeba gingivo, okay, or gingivalis, those are completely harmless and common cells. They are present in the body, they remain there and develop, but they are harmless. Whereas Entamoeba Hystolytica, it co-lives in human cecum and colon and is non-injurious common cell initially, remember, but later in certain condition it become virulent pathogen and it may cause a disease. That disease is called as amoebiasis. Okay, so this is about the systematic position. Now where you can find them, Entamoeba Hystolytica, where you can find them. So if you look at the geographical distribution of Entamoeba Hystolytica, it was initially discovered by Lambel in 1859 and its pathogenic nature was demonstrated by Losch in 1875. Animal has a worldwide distribution, but more common if you look at tropical region. In previous topic also, the Plasmodium vivax, we have talked about that their distribution, and there also we have seen that most of the time they are common or present in tropical and subtropical region. So our tropical region and subtropical region includes the India, subcontinental, sub-Sahara Africa, some parts of Africa, not everywhere. Okay, in China it is there, Australia as you can see, in Brazil, Madagascar, Mexico is there, and some part of USA it is also present. So basically they are present in tropical region. not every. If you look at the Canada or north side of America, you may not see this. Even the dry areas of Africa, in Sahara region, there also you don't show this or see this, because it's a dry region. So basically, wherever you have water, that is the main area where survival takes place. So this is the geographical indication of Entamoeba Hystolytica. Now, what are the hosts where it occurs? Now, as I have already told you that basically in humans they are present, all those 3 species, but if we will talk about only the Entamoeba Hystolytica, so it is present or found in human beings first, but often they can be found in the apes and monkeys, and it may also occur in dogs and even in wild rats also. So they are present in these different hosts, but main hosts are the human beings, and experimentally it is possible to infect kittens, hamster, guinea pigs, white rats with this parasite. Why it is experimentally? The reason is, if you want to study them, you have to infect them to some animals. Now, as you can see, guinea pigs and rats or hamsters, or sometimes kittens also, not kittens but hamsters, guinea pig and rats, white rat, these are the lab animals, and by infecting them you can observe or you can do different experimentation related with the Entamoeba Hystolytica and their effects within the intestine of these animals. So from there you can find out what can happen in humans also. So that's why experimentally it is possible. Other than that, if we talk about its habit and habitat, then it's a obligate anaerobe, required temperature. Obligate means it is not a strict anaerobe. Anaerobe means those organisms which does not require oxygen. It's a obligate anaerobe means even in presence of oxygen it may survive, it will not die. Those which are strictly anaerobic, they die because they cannot survive in the presence of oxygen, aerobic condition. So it's a obligate anaerobe. It is require a temperature range of 32 to 41 degrees Celsius, which is and in nearly neutral environment. Neutral environment means not extreme pH and extreme alkalinity. 32 to 41 degrees Celsius, it's a general temperature that can be found in most of the mammals. So that's why they are not found in reptiles. Reptiles, these are cold blooded, means their temperature may vary in a very long range. Because of this reason, you may not find Entamoeba Hystolytica infecting some of the reptiles or any other species. Most of the time mammals in fact, reason is body temperature of most of the mammals are constant, and that too in the range 32 to 41 degrees Celsius. Our human is 37 degrees Celsius, and if we talk about the environment where they can survive, it is 6.5 to 7.5 pH range. Now this range you may see the 1 or 2 pH in the stomach, but once they enter into the intestine, it is alkaline, but still 6.5 to 7.5, that is the range. And also they may found in the sometimes in blood, whose pH is also neutral. So this is the conditions which are met with the human intestine. The name Entamoeba is indicative of this habitat. Remember, now look at the name Entamoeba. Amoeba enteron means amoeba. It's belong to that species amoebic, but where they are present inside, that is enteron or intestine. So they are adapted for the intestine of any animal. So normally protozoan is restricted to a large intestine where it lives as a scavenger. Remember, scavenger means whatever dead and decaying, or we in general terms, scavenger means any animal which feeds on the dead and decaying material. Okay, they do not kill anything. Whatever it is there in front of them, they eat. Now, in our body, in intestine, we don't have dead and decaying material actually, but we have the semi-digested material. So it involves formation of vacuoles by invagination of plasma membrane. Okay, and it feeds upon what? Feeds upon bacteria and similar size decaying food particle and dissolved macromolecules. So within our intestine, if we talk about, then within our intestine we have the food material, semi-digested food material, macromolecules are there, semi-digested food particles are there. All these things can be taken up by the entamoeba. This is the amoebic one. So with the help of pseudopodium, it engulfs the macromolecules, and along with this they also eat the bacteria. In our intestine we do have bacteria. You have heard about Escherichia coli, and other than that there are so many bacteria which are present in our intestinal region. Okay, so those bacteria, they feed upon those bacteria. Occasionally parasite may venture into lower part of small intestine, okay, in sea of foods. Okay, so this is about the habit and habitat of this particular animal. Sometimes the solutes are also taken up by the pinocytosis. Solutes are there which has taken up by the pinocytosis. Pinocytosis is the process, or we can say the drinking by the cell. So in that, the large vacuole is not formed, very small pockets of vacuoles are formed. That is called as a pinocytosis. Inside they take it. Okay, so occasionally parasite may venture into the lower part. So this is how it takes place. Now other than this, the feeding, okay, or growing stage of Entamoeba Hystolytica is also known as trophozoite. Now here, from here onwards, you should know about like that we have seen in the Plasmodium vivax, that from entry in the humans, and when it goes back to the mosquito, every time, every stage they have a separate name. Similarly, here also there are some stages. So there are separate names for these stages. So feeding or growing stage of Hystolytica is called as trophozoite. So when it enters into our body, whether it's a trophozoite, not necessary, its name will be different, that we will talk about. So first of all, trophozoite. Greek tropho means nourishment, zoon means animal. So the stage where the nourishment takes place, that is called as a trophozoite stage, and this trophozoite stage exists in two forms. First, minuta, remember, in gut and lumen, minuta, it is present in gut and lumen, and magna, this is found in tissues. In some of the minuta form grow to be magna form. So from minuta, see, minuta means it's minute, small, and magna means a large one. Okay, so magna form trophozoite may also enter into the blood vessel and reach organs such as liver. How it reaches the organ, that we will see in the life cycle later. So finally the encysted, it can goes into the liver, lung, brain, and even when they start eating tissues, they may go into the brain. We have already talked about one of the amoeba, which is a brain eating amoeba. Okay, so the encysted E. histolytica may live outside the host body. So how exactly they go outside and how others are getting infected by this particular part? So in that case, when it goes outside, they are in the form of cyst, okay, encysted form, and they outside the body in the sewage they may be present for about a month, but can be killed by the temperature 50 degrees Celsius. Sometimes the sunlight, if they are dried out, this encysted part will they become dead, or sometimes desiccation or extended period of time in chlorinated water can also kill them. Okay, so it is possible that parasite is capable of lysing tissue cells. So before encystation, they when they are in the magna form, what they do, they live in the mucosal crypts and submucosa of the large intestine and start eating those tissue cells. Okay, and an ability reflected in this specific name, that's why it is called a histolytica. Histo means what? Which literally means tissue dissolving or tissue eating. Histolytica. It is called as Entamoeba Hystolytica. See, we have one more Entamoeba coli. Okay, coli means it is present in colon region, but it is harmful? No, it is not harmful. But here Entamoeba Hystolytica means amoeba, entamoeba, which can eat the tissues. That's why the name Hystolytica. Now how the locomotion of this takes place. So locomotion of Entamoeba Hystolytica can be observed in a worm. Okay, smear of fresh stools, 37 degrees Celsius. Now how they move? The movement is not. It's like a jerky manner. They push themselves. Okay, they don't have a proper cilia like movement. Slow movement, jerky movement. Okay, with a single large clear implant ectoplasmic pseudopodium. So also sometimes it is called as the lobopodium. So everywhere this characteristic part of this particular Entamoeba Hystolytica, as you can see in the diagram, lobopodium, okay, which is projected in an explosive fashion in direction of the movement. So it forms when the movement takes place. Protrusion extends, the rest of the protoplasm flows into it. Okay, so there will be a protrusion first in front, and then rest of the cytoplasm will go towards the protrusion. So it become again spherical or circular. Again the protrusion will form in front, and again it will go back normal. This is how the movement takes place. So now let's talk about its morphology. Okay, in the life of Entamoeba Hystolytica exist in three distinct morphology, morphological or physiological condition. The minuta form, trophozoite, then you have magna form, and the third one is the cyst stage. So if you look at the diagram carefully, I'll try to enlarge this diagram. So as you can see, the minuta form, in this you see the digestive vacuole with the bacterium. There are many things. We will talk about one by one about the minuta first, then the magna. As you can see, lobopodium is there, nucleus, very distinct nucleus with nucleolus is also you can see, and there are some digestive vacuoles with erythrocyte. So basic difference, if you look at it, minuta will have the digestive vacuoles with the food particle or bacteria or some other part, but you may not see any of the digestive vacuole contain the erythrocytes or part of tissues. Whereas if you look at the magna, then magna you will not see any digestive vacuoles containing the bacterium or any other food material, whereas you will see the digestive vacuole with the erythrocytes or tissues. Okay, so this is the basic difference. We will talk about the minuta first. What are the things present in the minuta? So minuta form trophozoite. This is actively feeding and growing stage, found only in intestinal element close to the mucosal surface. Remember, minuta form is means it is not harmful in the body unless and until this particular trophozoite or this minuta is there in intestine. It is not harmful because it's a scavenger. It will just feed on whatever it is present in the intestine, not on the tissues. So cell body, it's a typical cell, a single cell. Okay, delicate plasma membrane is roughly oval in shape, and has size 10 to 18 micrometer. The cytoplasm consists of thick band of clear gel-like peripheral ectoplasm or hypoplasm. Okay, and finely granular is there. Then hypoplasm contain microfilaments. Microfilaments are the parts of the exoskeleton, not exo, sorry, they are the part of the cytoskeleton and sharply differentiated only toward the pseudopodial region. So this movement of pseudopodia, that is because of the presence of microfilament. Then what else is there in the minuta? Further, in symptomless host, remember symptom means what? The host is not having any problem even though it is infected. Trophozoite rarely shows an erythrocyte containing food. As I have told you, trophozoite generally, minuta form, generally don't eat the tissues. Okay, most of the food vacuoles are fluid filled, although some may contain ingested bacteria and cell debris. Now there is lack of mitochondria. Remember, this is a very distinct thing. Golgi bodies and ribosomes are present, but rough endoplasmic reticulum has not been seen. First. Second, there is a total lack of mitochondria. So even though it is a typical cell, unicellular organism, you may not see mitochondria. What is the reason? We will talk about that reason. Numerous glycogen granules are scattered in the endoplasm individually or in clumps, and round vascular nucleus is located in the granuloplasm. Okay, 3 to 4 micrometer in diameter. Small central densely stained granular endosome or karyosome is also visible in the diagram, as you can see, composed of condensed DNA. So a very important thing, clumps of chromatin rich in RNA forms a dense layer close to the double membranous nuclear envelope. Okay, now the endosome is often surrounded by clear halo, and sometimes spoke like lines may radiate from the endosome. Okay, so why this is all about the minuta, and why it lacks mitochondria, that we will discuss when we will see the life cycle, because this is part of life cycle. At that time you will understand why it doesn't have the mitochondria and why you have the glycogen granules. Now we will talk about the magna, next stage. Now in magna form, trophozoite, it is a large as compared to the minuta, produced mitotic division and growth of some of the minuta form trophozoite. Okay, magna form distinctly larger than the minuta. How much? The previous one was nearly 10 micrometer, whereas here it is 20 to 40. It's a mistake, sorry, it is 20 to 40 micrometer, not meter. It's my mistake. The nucleus is also somewhat bigger, which is 56 micrometer. Okay, and there is no other cytomorphological difference between minuta. Other than this, you don't see anything. Large size is there. Okay, and large size of magna form may be related to the nutritional richness of the environment, because now it is feeding on intestinal mucosa and submucosa and other tissues. Okay, and sometimes if from here it enters into the blood, and from within the blood it will feed upon the erythrocyte, it will go towards the liver, it will start feeding on the hepatocyte, it goes towards the brain, it may start feeding on the brain cells. Brain it is rare, but still. Okay, so from once it entered into the blood, it will go anywhere and start feeding. Other than this, minuta form, as I have told you, it is restricted to intestinal lumen. It doesn't go anywhere. So this part, magna, it's a tissue dwelling. Okay, nature and cytophagic habitat. So magna form trophozoite is characterized by presence of 1 to 10, sometimes 40 endoplasmic food vacuoles. So there will be lot of difference. In minuta you may see food vacuoles, small food vacuoles, but not very with large number is not there. Whereas here magna form, you sometimes see up to 40 food vacuoles. Okay, which may contain erythrocyte undergoing digestion. So organism does not feed on bacteria, remember. Also, the magna form trophozoite uses the method pseudopodial capture to engulf erythrocyte and other cells like hepatocyte. So food vacuole thus form known as phagosomes. Okay. So this is about the magna. Now the third stage, the third and the last stage. After that we will see the life cycle. So cyst stage. Now after magna, okay, the product of only lumen dwelling trophozoites found only normal form feces and never in the dysenteric or liquid stool in the tissues. So where they are found, cyst stage is found in the fecal matters and not in the dysenteric or liquid stool in the tissues, remember. So after period feeding, reproduction, many mature minutiform trophozoite enter this stage. Now this immediate stimuli for encystation not known, this is unknown. Still I have searched some of the papers, but the reason is still not that much known. But glucose starvation may one cause, because cyst stage in any of the amoebic part, there is one thing that cyst stage occurs when you have a lack of food, but here they create such conditions so that they should go into the encysted stage. So there are three phases: precyst, cyst and metacyst. So in precyst condition, the uninucleated minuta form, see the diagram is there. Precyst condition, what thing happens in the precyst to cyst and then to metacyst. So in precyst condition, uninucleated minuta form seizing, moving or digest the remaining food, okay, shrinks the volume and begin surround itself. So what it will does, it will stop the food material. The number of vacuoles will be reduced, its size will shrink and it start surrounding itself with a delicate single cyst wall, okay, which latter become thick. So initially it is not thick, later it become thick, because later it has to go outside and protect itself from the outer harsh condition. That's why cyst wall formation takes place. Now in precyst, further characterized by single nucleus, mass glycogen near further, and one more refractile chromatid body is present. See, glycogen mass, a large glycogen mass which is even larger than the nucleus sometimes, and chromatoid body are gradually developed into the cytoplasm. Now these are the ribonucleoprotein, remember the chromatoid body. Ribonucleoprotein are also called as RNP and appear along the body rounded ends, okay. Now cyst wall now become refractile, gradually disappears and metabolized. Now stage advanced from here onwards, precyst, it goes towards the cyst. So as you can see, in precyst you don't see the wall, whereas in cyst and metacyst you can see the wall, thick wall. There is a change, glycogen is there, but now it is smaller in size, okay. So the spherical cyst about diameter show 2 nuclei. So you may see the 2 nuclei. Now glycogen is reduced, okay. After this there will be last maturation phase, that is the metacyst. Marked presence nuclei formed by division. So nuclei, previous phase it was 2. In cyst, see, precyst you have 1 nucleus, in cyst you have 2 nucleus, whereas in metacyst you will see the presence of 4 nucleus, okay. And this is the last stage. So when the precyst stained with the iodine appears dark brown, okay, must chromatid body unstained. Now this however can be visualized, dark body stained with the iron hematoxylate. Now disappearance is more apparent than real, the RNP merely become dispersed. Now rarely does metacyst contain 8 the nuclei found, somewhat smaller, okay, 4 or 8, 4 is there, okay. In quadrinucleated metacyst passed out of the host body, remember this metacyst passed out of the host body and become infective when ingested by man. Now, cyst form enable these parasites to resist the effect of unfavorable environmental condition outside the host body, and once this cyst enters into the human through the stomach, okay, at that time this cyst wall, it protects the metacyst from the digestive juices, unless and until, so it protects, and you will it will go towards the intestine where this particular cyst wall will open up. That we will see in the life cycle. Okay, so this was about the initial habit and habitat of Entamoeba histolytica. In next lecture we will talk about the life cycle. Thank you.